Hi, I'm Jennifer Ouellette, and I am a blogger with Cocktail Party Physics, and welcome to Science Saturday, and I am talking with...
Ricardo Gil da Costa. Hi. Hi, Jennifer.
Hey, how are you? And Ricardo, maybe you could tell people a little bit about who you are.
Sure. So I'm essentially these days a cognitive neuroscientist. I'm a researcher at the Salk Institute for Biological Studies in La Jolla, California. And, anyway, I work on mostly on brain imaging. So I'm looking at the evolution of language and how do people do conceptual representation. So how do we represent different types of objects and events in our brain? What's the behavioral part of it, and what are the neural bases for it? So that's what I do these days.
Okay. And we're going to actually talk quite a bit. This is going to be kind of a brain-heavy Science Saturday, but I can't really, let one thing go without mention, which is Large Hadron Collider had its first collisions earlier this week. People stayed up all night trying to get the live feed, and someone did mention that the irony, of course, is that they achieved this very big machine in the world's highest energy collisions, and yet they couldn't really get the live feed to stay online. But such is the irony of high energy physics. But we have to kind of celebrate that a little bit because it's a huge milestone for particle physics.
Absolutely.
Yeah, the LHC turned on. It's been decades in the coming. It turned on like a year and a half ago, and that was just it warming up. And this is like the first collisions. So now I think what's exciting about it is we're actually going to start seeing some actual results. It's kind of like when you finally get your experiment to work.
Exactly. Exactly. And it takes a long time, so at least it's very rewarding when you get to that part.
Exactly. This is your first time on Science Saturday, but I definitely want to talk a little bit about your background, because you have a really interesting story. You didn't just spring fully formed as a neuroscientist into the academic community.
No, not at all. It's been kind of a very twisted road to get there.
So maybe you could talk a little bit about this kind of roundabout way that got you into, studying language centers of the brain.
Sure. So actually, my background is in biology. So I graduated in biology, and then I moved to a master's in marine biology. And actually, what I was always interested since I was a kid, and I was a big Cousteau fan. So I was always very interested in marine biology. I grew up in Portugal, half the country's coast. And marine biology was really my main focus, and I was very interested in doing. So I worked on that. So I got the master's, worked on that for a couple of years. I worked actually for the European Union as a researcher in parts of Africa and Europe. So I was in Kenya, Tanzania, Swaziland, South Africa, doing some field work there. And then we did a lab work back in Portugal and in different European labs. And then I also started getting more interested in working with sharks. So I spent also some time at the Bimini Biological Field Station in the Bahamas that is affiliated with the University of Miami.
What was it about sharks that you found so fascinating?
Sorry?
What did you like about sharks?
Everything. Come on, they're fantastic. They're like amazing creatures. What is there not to like about sharks? They're very misunderstood though, but they're amazing. I mean, so it's different things, right? One of the really, for me, one of the really captivating things that is incredibly battery of sensory systems. That you don't see anywhere else, from a lateral line that you see in other fishes, but also to something that we call the bioelectric sensory system. So basically, close to the snout, they have sensory ampullae that can feel differences on the electromagnetic field on the water. And this has tremendous, crazy applications for them in the way that, for once, they can do what we call homing, so they can feel the Earth's geomagnetic field and feel their own electromagnetic field that is created by their own bodies. So when they're moving, it's like they have this automatic compass on their head. They'll always know exactly where they are just based on these two vectors, right? So that's pretty cool in itself. They get to hunt prey who can be buried anywhere, like under the sand. So they use that as a last thing for prey detection, together with an amazing olfactory sense, all sort of gustatory sense. So they're really amazing machines from just a sensory input and integration kind of aspect. And come on, they're cool,
Yeah, well, there's a reason Discovery Channel Shark Week is so delightful.
Exactly. And it's
really
important,
popular.
because with things like Jaws, which was a great movie, I definitely loved it when I was a kid. However, really give these tremendously bad reputations to sharks. Dolphins is the thing that everybody wants to love, and sharks is the thing that everybody likes to hate. And it's really unfair, actually. I mean, when I was in Bimini, we used to do different shark dives per day. We dive with tiger sharks, lemon sharks, Caribbean reef sharks, black tips, white tips. And we never have a problem. And people have been working on that station for decades. And it's very, very, very rare that we even have any kind of accident. So I think they've really misunderstood.
Well, I'm glad you're sticking up for the sharks.
Absolutely.
So you did shark-
anyway,
you did
I got
shark
interested
research.
in part of the electrophysiology and how basically how different animals integrate sensory information and into their brains and how they represent all the world, all this constellation of objects, of images, of events that they have in the world. But one thing that I felt at the time is that although I really like sharks as a model for other things, I started getting interested in more cognitive aspects. And for that, sharks are definitely not a great model. So one hypothesis will be going towards marine mammals, but in that sense, there was no really good non-invasive imaging technologies to look into their brains and to have this non-invasive approach of the whole brain about what's going on in vivo. So the other option was going back to either humans. I was very interested in the evolutionary approach as a biologist. So the other option was going to humans, but I needed to find another animal model that I thought was more suitable for cognition. And in that sense, because I've been working in different African countries for about 2 years doing the marine biology stuff, but I got acquainted and got to hang out and deal with a lot of people working in primatology. So I became very interested in that. So for my PhD, what I did was actually doing that, was doing behavioral work with different monkey species, both in the lab and in the field, and integrating that with non-invasive brain imaging studies. So we developed this setup to do positron emission tomography, which is this type of brain imaging, this technology that allows us to look at the brains in vivo and compare different tasks, different processes, and see which neural networks or which parts of the brain are relatively more activated for one versus another. So we did 2 different types, PET, positron emission tomography, and fMRI, which is functional resonance, magnetic resonance imaging, during my PhD. So that's what I did. I developed a setup where I could do this with using both human setup but adapted to animals, in this case specifically for primates, and do comparative studies with them. So it's kind of a winded road to get to neurosciences, but it sort of took me there.
Yeah, I was actually, I went down and visited you, obviously, at the Salk Institute and got to see your lab. And first of all, you have the world's tiniest office, and it made me feel much better about my tiny office.
Absolutely. And you know what? This is actually an improvement. I spent about 5 years at the NIH, at the National Institutes of Health in DC before that, and there was no windows. So now that I actually have a window where I actually see the ocean, I'm happy, even on a tiny office.
And the other thing, I remember us talking about the fact that you developed this really kind of innovative new brain imaging system, and people think that it's so easy. You should just be able to take these techniques. We've been using them on humans for a very long time, and you can just kind of throw those on an animal, and it's going to work just fine. But the monkeys have personalities, and they get scared, and there's certain, their heads are small, and there's all these challenges that you had to overcome. So maybe you could talk us a little bit about that.
Absolutely. I mean, you're very right. It's a big misconception how easy or trivial it would be to adapt something that we do in humans to other primates, different monkey species. In this case, we've been working with rhesus macaques, and it's really not true. They're not just tiny humans. There's a lot more to it. And one thing that I realized, during my PhD, doing the behavioral part, and I definitely adapted when I was creating the first setup at the NIH, and now this new one that I'm creating at the Salk, is that you really need to adapt a very strong training protocol, adapted to the personality of each of the animals, and use that in your behalf to kind of get him to what you want him to do, which is, in this case, is actually just laying very passively in the scanner, just like a human subject would do. But there's different aspects of it. There's this behavioral part. There's the question about just the physiology. For instance, in the case of monkeys, they have this very thick cranial muscle that interferes with some of the signals for some of the recordings that we want to do. The skull shape is different, so we have to adapt head coils, and in this case, so one of the additions that I'm actually doing now with the Salk Institute is, because of course we're never happy when you resolve a problem and you think, instead of actually milking that, no, we decided to add more problems. So one of the really great things that we got going at the NIH is that we have a very good spatial resolution about the different areas of the whole brain to examine different functions, but the temporal resolution is very poor. And most of these cognitive and neural brain processes are extremely fast. So we wanted to have another technique that would have a better temporal resolution, and that's EEG, or electroencephalography, which again is very commonly used in humans, but there was not a good monkey setup to do that. So you have to create our own EEG cap. So it's basically an elastic cap that goes over your head with different electrodes on that. So that's what we have for humans, and we created a similar thing for monkeys. So to adapt all these systems so that we can have the simultaneous fMRI that gives us this big spatial resolution with electroencephalography that gives the temporal one. So all these things are a lot of the issues, like the devil is in the details, right? So it's how you're going to adapt things and to get them to work. And I can tell that we've been at the Salk right now. I've been here for about 3 years, and only now we're starting to get the system to work. So it's definitely far from trivial.
Right. Now, I just remember, seeing one of the little monkeys and you telling me that, one in particular doesn't like to sit still at all, and she won't. She won't be calm unless someone is actually actively holding her hand.
Right. So
and that's
really,
just a personality thing.
Exactly.
Monkeys
it's really personally adaptive, right? It's one of the females that we had. A male, the one that we have, don't even try to hold anything, don't get close to holding his hand. He's certainly not going to be happy about it. So
he has intimacy issues.
Exactly. So you really need to adapt to different animals, all of your different training, and it's very important that you take that in consideration. I think actually probably a lot of the experiments, or part of them at least, that weren't successful in the past is because we weren't very aware of this, and we tried to do it in this sort of standardized way, which turns out to not be the most efficient one.
Yeah, I have just a brief segue before we keep going, because obviously, a few months ago, a couple months ago now, there was a seminar at UCLA. Animal rights activists get very, very upset at any kind of animal testing, and you are, in fact, using live monkeys. And so I'm curious as to whether you've had trouble with that.
Personally, directly, I never did, luckily, which is very good. I know that I'm mentioned in different animal rights activist sites. Actually, some don't like me, and they don't like any researcher who does work in animals. But actually, it's funny enough that some of them seem to like me. And the reason for that is in some of the articles that we published, we really made a point about how the conceptual system, so the way that we humans represent different concepts, different ideas, different objects, is actually very similar to the way that monkeys do. Of course, at a whole different level of the language, but the core structure at the behavioral neural base are very similar. And they felt that would, give a general public an idea of, how close we are cognitively to these animals, and that would increase the respect towards the animals, which we really hope that it will. I mean, it's a big part of it, right? So it's sort of a controversial thing. I never actually had a situation specifically directed at me, but I can tell when I was starting at Harvard, when I did my PhD at Harvard before moving to the NIH, just on the first day that I got to the lab, we had, Harvard police there, Boston police there, because there had been threats toward the PI at the lab at the time. So it's a very tricky situation. I do have colleagues who personally have very serious problems about this, including their families being put in jeopardy, being threatened, their kids being threatened out of school, destruction of their property, cars, houses. It's insane. It's out of control. And I mean, it makes no sense. It's crucial that you have a rational argument about this. And obviously it's important to keep the animals' rights in perspective. It's important to give them the best possible conditions to do this, regulate. And we do have very strong boards to regulate how animal rights are enforced and how animal research is done. From the number of animals used to which species you are using, if it's really necessary, to give them the best possible living conditions and all that. And that's what you should be focusing about, instead of going to two-way over radical ideas that are really not applicable and ended up being a form of terrorism in the same way, so which is simply not acceptable, obviously. But then one of the problems that I think some of these associations are very good and they're well built, but others are not, and you probably get the wrong people on it. And then it becomes a problem, because sometimes they even show an extreme amount of ignorance about the subjects they are trying to protect, so which leads to misleading information and all sort of added problems.
Right. So you mentioned during that something about the way that the cognition processes and the languages of monkeys. I mean, it's not language the way we're normally used to thinking of it, but I remember you telling me that you were learning something very interesting about the root of where language comes from, and whether it's in pictures or whether it's words, or maybe you could expound a little bit on that.
Right. So some of the work that we did was actually, as other colleagues did, that we did at the NIH and we're continuing now, was actually looking how, so monkeys, different monkey species, in this case rhesus macaques, different macaque species, have a very rich vocal repertoire of about 10 to 12, depending on the species, sometimes less, but in this case of rhesus macaques, about 10 different types of vocalizations. Vocalizations who have a meaningful content, in the sense that when they're giving a very specific social context, like an affiliative positive approximation or a threat, or when food is discovered, or when a predator is present. And each category of these vocalizations is giving that specific context, and does elicit a specific behavior. So, for instance, an eagle alarm call, which is a specific thing in vervet monkeys, for instance, will get the animals to go and hide under the bushes, as a leopard alarm call will get them to go climb the highest possible tree to escape. So the responses are very selective and very specific about the type of information that is contained. The same thing, for low-level foods to high-ranking rare foods, so that there's an incredible richness in their vocal repertoire, even though very limited, obviously, in comparison to ours. And even though, as far as we know anyway, they obviously don't have language per se as such, right? One of the crucial things about language is the capacity for recursion. So the fact that with a finite set of elements, we can keep recombining them to have an infinite number of words, an infinite number of meanings that we can type them into, and so forth. They don't have this kind of computational recursion processes in any other species as far as we know. That is not ours for that. However, they do already have this very rich mental representation world that is already expressed on a vocal communication system with different meanings for different vocalizations. And that does seem to be one of the building blocks to get to the point where we are with then our language totally broke free from that with recursion and with the capacity to actually, because we can construct these long sentences, we can talk about events on the past, we can make predictions about events in the future, and in their cases, it's in the moment. The vocalizations are given in a specific context, so it's always in the present. However, it does denote, like I said, a very rich mental world that does seem to be at the basic level of conceptual representation, and is one of the building steps for us for language, for what they call the semantics part, this dictionary of all the meanings. And another very interesting thing is that what we found out is when we look at the neural network, means that at the ensemble of brain areas involved in this, the homologous brain areas in the macaque, so the identical brain areas, are the one that gets selectively activated when they're processing these conspecific vocalizations of their own calls, is homologous, is identical to the ones that humans activate to process language. So one of the interesting things as well is that, so the way that we do this conceptual representation, right? So when we are acquiring an experience about something, let's say, for instance, a dog, right? If the first time that you experience a dog, you're going to create a memory of this dog. But this memory is going to have different attributes. Like, you're going to remember the shape, you're going to remember the color, maybe the texture of the fur, a barking sound associated with the animal. So there you have visual components, you have auditory components, the smell of the animal, so you have an olfactory component, and even an emotional valence associated. Let's say that you really like the dog or you're really afraid of the dog. And the way that it seems to be represented in your brain is spread. It's not like one single memory put in a specific brain area, but instead, there's actually all these attributes are spread in a broad cortical area. So visual areas, lower and high-order visual areas, visual cortex, is going to be responsible for storing, experiencing, and storing the memory of the visual attributes of this object that we encounter. The same thing for the auditory ones, the same thing for the olfactory, etc. And what happens is that when this memory gets elicited again, being because someone told you about the dog, or you hear the barking sound again, or whatsoever, all this sort of comes together. All these, the areas that get activated when we experience this, get activated again on the retrieval. So it's this group, this ensemble of areas that make emerge the representation of this dog. So, and this seems to be the same kind of neural network that we see for mental representation in the macaque. So it does seem to be an evolutionary continuous between the way that primate brains do this, obviously in a much more limited way in non-human primates, but it does seem to be a building block to what we as humans have right.
Right. It's interesting because I've just kind of started getting interested in brain research, because it's kind of exploding right now.
Yep.
But one of the things that's really changed is I think that for people who haven't kept up with it, we still kind of have this idea, that the brain works like files in a computer. You have left brain, you have right brain, and everything gets filed under the file named, marked dog, and it's not. It's much more diverse, and it's much more complicated, and the neural networks and the synapses are extremely complicated, and it's also extremely plastic, and it can rewire.
Exactly. I mean, it is extremely complicated. I think one of the big paradigmatic shifts that we had in the last 15 to 20 years, and moreover in the last 10 years, although it's still pretty much on public knowledge, you still thinking this way, is that the old idea of the brain divided into areas and this sort of almost one-to-one or one-to-two correlations between area A does task 1, area B does task 2, and whatsoever, it's really being put aside. Exactly as you're saying, the way that we look at it right now is that we have this combination of areas working together, orchestrated to perform a certain type of cognitive task. And then some of the areas on the group for task A are also going to be recruited for task B together with other areas. So it's all about these neural networks. It's all about these networks, these nodes of different areas, different cell populations work together to perform a certain task, being at the perceptual level, being, like sensory systems, being at cognitive level even more, to things like memory, language, sleep, etc. So it does seem to be a much more fluid and dynamic process, and much more less of a set of boxes, and then each part, it's part of the machine does one single function. It's much more complex with that. Also, the other thing that has been very hot topics these days is how this information is passing between different cells. We thought that most of the information, if not all of them, actually was based on neurons, and now there's other types of brain cells. They're part of what we call the glial cells, so part of the matrix where these neuron cells are embedded, that also seem to be, contributing to information. So it's an incredibly complex system, and to some extent we've been making tremendous progress, especially with the development of the possibility of these brain imaging methods, where we can actually assess in vivo while, on different subjects, while this process is, being a linguistic task, being a memory task, being just a perceptual task while it's happening, instead of just using post-mortem autopsies on brain lesion patients like we did, 50 years ago. However, for me, at least personally, I keep feeling that we have these fantastic insights, but it's almost like the more that we know, the more that we understand that we know very little about the brain. And it's still a big mystery, probably one of the biggest mysteries that we still have so far.
Well, that kind of, I know that you and I have talked about, fMRI imaging, in the past, and, that's certainly, the images are stunning. Earlier this year, I actually participated in a brain mapping study. Last year, I participated in a brain mapping study at UCLA, because apparently, they're still using the old brain map from some woman in, the 1970s.
Yeah.
And so UCLA is essentially conducting this very broad study with, hundreds of subjects of all ages, and we all go in and we perform cognitive tasks, and it images our brains, and then it's basically going to try and find the common areas and come up with sort of a composite brain map that'll be a little more accurate to guide surgeons and so forth. And the images are stunning. They showed me pictures of my brain, and it was beautiful. But people sometimes attach too much meaning to the fMRI. We are told, in news article after news article that we can read minds and we can tell if you're lying and you can, do all this sort of amazing stuff. But it kind of gets back to what you were saying of, every time we think we know something, we realize how little we actually know. And a lot of those images are very much open to interpretation, so it's kind of tricky, right? Right.
It's extremely tricky, really. And one of the issues that I think that typically is misunderstood at a public level, because of the way that it's portrayed and put outside, is exactly how do these images get created and get together, right? So one of the things that intuitively, when we see a picture, when we see what looks like a photo, we think, that's our visual senses telling you that's real. I'm looking at it, I'm seeing it. But in truth, the way that we create these images, that there's two types of MRI images. There are the structural ones, which are really, just about the anatomy of your skull, of your brain, muscle tissues, etc. And those, it's really just a type of photography, at lack of a better word.
There's one big nerve that they imaged on mine, and it looked very, very pretty.
Right. So the structural part, it's obviously with a different method, but it kind of works like a photo. But the functional part does not. So when we see these patterns, these colorful patterns that, like you said, are very popular these days, from TV shows to newspapers when they want to, put some of the discoveries out, it's great to have these beautiful images of a brain with these different color spots that show the activation areas. Well, in a lot of cases, and this happened to me both in public conferences, in public lectures, and also actually even at undergraduate students level, that they actually think that the color part, so the functional part, actually also comes in the same process as the structural one. So it's kind of, a photo of something, which could not be further from the truth. The whole functional part is actually a statistical construction. So what we do is basically, we compare 1 to 10 tasks, but just in the sake of simplicity, let's say task A and task B, right? So we put our subjects in the scanner, and ask him to process task A, and then we do the same thing with B, and we compare one against the other. And when we compare it, we basically do, well, you're the worst one on calculus and statistics, but when you compare it, it's basically a t-student test in most cases, or whatever statistical analysis that a researcher decided to use to compare voxel by voxel. And I should say that a voxel is like a three-dimensional pixel. So if you think about a pixel like an image on your TV, it's a three-dimensional cube of that image, and that's what composes the image of a brain. So each voxel is going to have a statistical value when you compare these two conditions. And basically what we see is what we call the brain maps, or the T-maps, these activation maps that are colors. The colors are just relating a value, a statistical value of comparison between these two conditions. So really, it's instead of having a ton of tables with numbers, we create this much more pleasant and much more efficient way to visualize where we find differences between the processing of two tasks for a given area by doing it in this visual representation. But it is a construction. It's nothing but a statistical construction. And so, of course, having that said, it's completely dependent in the way that we design the paradigm, in the way that as scientists we tested the whole process, and the way that we analyze it. So they're not more or less real than any other mathematical operation. That's what it is. So it's very tricky. So sometimes, depending on how people more carefully or less carefully constructed their research paradigms, their research experiences, it can be very reliable or less reliable. But by no means is ever just a picture, and therefore it's a photo, so we're seeing it, so it's there. It's there because we put it there as a statistical construct, right, and nothing else. Now, on top of that, there's also, like you said, the whole issue about now how we're going to, even if it's coherent and we do understand the analysis that we're doing, how do we interpret certain type of results, and that ties together to how much we know and how much we don't know about brain and the brain processes. But I think one very important thing is just sort of try to clear the myth that, when you look at these brain pictures, like a picture is like a photo, so if you see it, it's there, and really for the public to understand that that is a construct.
Right. Getting back to the, we can read your brain and tell if you're lying. I think it's the same problem as with any lie detection. It's lie detection, the standard lie detection equipment has pretty much been debunked over and over again because it's accurately measuring certain bodily responses, but it's making certain assumptions about what those mean.
Exactly.
And I think it was the first or second episode of Lie to Me, where they were showing off a new handheld lie detector, and Cal Lightman's character comes in and basically takes out the guy who's been asking the test questions and sends in a very scantily clad, very attractive woman to seductively lean over and ask the male subject all these questions, and he fails the test, even though he's giving the exact same answers he gave before, because his physical responses...
Of course, the whole physiological response has changed.
Right,
now he's aroused because of the presence of the woman, and that's going to completely shift the test.
So, does that also happen with the brain? Because the fMRI is measuring oxygen levels in the blood or the brain, correct?
Correct.
Because that's where increased activity is. And we make certain assumptions about what those mean.
Right, it does. You're absolutely right. And you even take it to even more indirect assumptions on top of those. So you're absolutely right. I remember seeing when they were trying to sell some of, or to promote these brain lie detection devices, saying, people lie, but the brain doesn't lie. Well, yes, the brain doesn't lie per se, but what we're doing is interpreting what we as people understand that the brain is doing at a given point. Usually there's two major types of so-called lie detection for the brain. One is based on fMRI, and the other one is based on EEG, on electroencephalography. But in a general term, they both come from the same premise, which is they're trying to compare, real memories by false memories. And the idea is that to some extent, you can assess if something that you're showing the subject or you're discussing with your subject, it's something that he has experienced before and therefore is a real memory, or it's something that he has no memory of, so he's making something up. And you should be able to distinguish that. And the idea is that in both cases that you can distinguish between the two neural networks that do this, creativity and therefore the lying, where they're confabulating about something, versus the neural networks associated with representing memories, so you already experienced them and you live them. But it's very tricky, it's very controversial to say the least, and I don't think it's ready to be accepted as such, because as you said, it's very interesting and gives us great insights in a very controlled laboratory setting with very specific paradigms. But when you take that and export it in the so-called real world and put this with much less constraint, then no, because whatever kind of results that you're going to have can be influenced for 10 other factors that you're not taking into account that can be doing that. So like you said, you can have this completely misreading of aspects, maybe in a more sophisticated way that we did with the old polygraphs, but suffering from the same general shortcomings, I think, exactly like you said.
Right. And this, I'm sure, drove you crazy. A couple of weeks ago, there was an episode of House where they had this hot new brain imaging technique where they were actually able to go into this girl's mind and kind of recreate an image. And at least they had the one character saying, "Oh, come on."
Exactly.
But the image
I was
that
very
came out
happy
from
about
reading her
that.
brain looked like a photograph. It looked like some sort of like night vision photograph. And I mean, I realize it's Hollywood and we take liberties and that sort of thing, but I mean,
well, we both know that they took a lot of liberties. But, we've both been there. But yeah, I mean, as a viewer, I'm a great fan of House and of Lie to Me. Actually, it's some of my favorite shows are those, like we talked before. But you're right, yeah, that episode drives me crazy. And it's funny because I usually don't like that character, but on that episode, I really like the character that keep complaining about all this. Because, and at least, I think something that was very smart of them to do in the process of writing it was saying that they tried to transfer to the TV show the controversy that we're living right now on the field about these kind of methods, which I think is very good. So at least we felt that there was representation of both sides, which is great because in most cases they don't even do that. But yeah, I mean, so one of the things that has been done, and that part is very interesting and is a big process, is doing what we call classifiers. So in a very general way, the idea is that if I sit you in a scanner, right, and for quite a while I showed you, let's say, 100 pictures of cars, and I have the computer program reading the types of brain patterns of activation that you have while looking at cars. Then I do the same thing and I show you some pictures of 100 pictures of animals, right? And then I repeat the same process with saying 100 pictures of foods. What basically, statistically, the computer program is going to do is seeing what is common between, the brain patterns of activation for each one of these 100 pictures of cars that you see, right? And the same thing for the animals and the same thing for the food, and then what's different between them. And with that, it's going to classify 3 types of categories. So one thing that we can do is, in fact, if you train these learning systems, what we can do is then potentially I could be showing you a picture that I don't know what it is, and just based on your pattern of brain activations, my program should be able to tell me it's a car, it's a piece of food, or it's an animal, because it basically created templates of brain activations previously for each one of these 3 classes. But then the thing is that then people think that based on this we can do it with anything. No, we can do it in a very specific conditioned number of classes, which is exactly the same ones that we train the program to do so. And it's not even true that we can say, well, in that case, if you set the subject and you train the program for 1,000 different things, you can read 1,000 different things. But even then, even though it's obviously not practical, but it's not even true, because then you might start having so much overlapping between the categories as far as the brain activations of them, and we don't really know what we're looking for, then even then we might not be able to distinguish anything. So we're making great progress, but it's at a very limited scale, and it's just almost like these very small sets, and try to recognize which one of these things do. So other people have been doing the same kind of approach, but instead of fMRI, they do EEG. So again, they're looking at the electrical waves that come from our brain, and now they respond to each one of these types of things, and again create these classifiers. But that's all they can do, and it's a big mistake to jump from that to saying, oh, so based on this, if I can figure out based on your brain activation patterns what you're looking at or what you're thinking about, I can extract this information from your brain. We're very, very far from it, if we'll ever even get there. But definitely not to this point. And then there's like commercial devices that are kind of cute and fun for like gamers to play games that will tell you that, have this new EEG system that is going to read your electrical activity, and based on that, it's going to read your emotions and what you're feeling about the game, and it's going to have this feedback, this positive or negative feedback with the game going on at the same time. Well, the truth is, in most of these systems, what they're actually doing, although they have these electrodes and they look at it, measuring your brain activity, what they're really doing is measuring different muscle contractions. And because typically, and that actually goes to the whole lie to me aspect of it, typically we have facial expressions that are directly related to a certain emotional stage or emotional experience that we're living. So what they're actually doing is using the different muscle contractions to infer your emotional state to whatever you're playing and use that as a feedback. So they give you this cool idea they're reading your brain, but actually sort of cheating out of it.
Yeah, they're using micro-expressions of sorts.
Yeah, pretty much. And in most cases, actually just
aspect,
macro-expressions.
I do appreciate House, though, because they actually do try, to bring in some of these new technologies and build those into the plot. And I do know, I remember one episode where they had someone who couldn't move because of something that was going on with his condition.
Locked-in syndrome?
I don't remember what it was, but they used they had him use the device where you can think and move a cursor with your thoughts.
Exactly.
And what I liked about it is they made it very clear that he couldn't just like automatically think. He had to train the machine, and it took him hours, to finally get this machine to actually be able to move that cursor. He could only move it up or down, yes or no. So it was like 20 questions.
So actually, that's for me. I mean, it's one of. There's a few of them, but it's one of the most striking and fascinating technological advances that we have done together with technology and neuroscience research. Is that actually works, and it's been an incredible breakthrough for locked-in patients. So, like we're describing, so we have patients that are literally fully conscious and alert, but it's sort of prisoners on their own body. And in most cases, they cannot move anything, and sometimes they can just do eye blinks or move slightly the eye. So exactly what we're saying, we're going back to the whole idea of training the machine, but then by training on basically moving up, moving left, moving down, moving right, you can then, have them controlling the way the cursor goes on the screen. So they start to do that, but we actually already have patients that are doing one of the virtual keyboards on the screen. They basically move the cursor to touch each one of the letters that come and, and do entire sentences based on that. Actually, that's a beautiful book, and they made a movie out of it called, trying to remember the title in English, I believe is the Bell and the Butterfly.
The Diving Bell and the Butterfly.
Diving Bell and Butterfly, exactly. Which is exactly one of these cases. And each day we're making more progress in the way we adapt EEG to do this. And that's one of the fantastic examples, I think, that is, recently being done that actually is working efficiently. That and a lot of the other parts is more motor control on neural motor prosthetics. We've been having a tremendous level of advances as well.
Right. You've actually, the reason that we know each other is because you've consulted for the Science and Entertainment Exchange, and I'm the director of that. And you know as well as I do that, as much as we try and get good science in there, they're trying to tell a story. So maybe you could share a couple of your experiences, about how you actually... They ask different questions. A lot of scientists are surprised. I mean, they're used to talking about their research to journalists and that sort of thing, but they're not used to being asked, well, what would that look like? And what would that person eat? And what would they wear? And, and where would they work? And that sort of thing. And all these practical questions. But they also, when they talk about the research, they want to know, well, can I do this with it? How about this? So how do you deal with the very different needs of storytelling?
Exactly. I mean, it's for me personally, and you know this, we contacted each other. Well, I actually contacted you when you started the Science and Entertainment Exchange, which, by the way, you're doing an excellent job. And I like to say that every time because it's very true. But in any case, so I was very interested personally in the whole, process of creating characters and entertainment, not just science fiction, but just fiction in general and drama, and how that translates, to the TV screen or to a movie. And so I was interested in getting involved with this as a science consultant. And I think it's a tricky process, and it varies a lot, as you know, right? Depending on the producers, depending on the writers, depending on the type of show or the type of movie, it's going to vary a lot. So we go from things that, are very, specific, like, they have a couple of questions that they want to have very specific answers for, and you can do that type of science consulting. So, for instance, for Fringe, which, as you know, is one of the shows that I've been collaborating with, typically is more on this format, is they have specific questions for any given episode.
Right, and actually my favorite quote by Glenn Whitman, who's on Fringe when he was at Comic-Con, he says, here's what our day is like. Our showrunner comes in and says, I need exploding heads. Heads need to explode. Figure it out. And then that's when they call you. And what do you do?
Exactly. And then my head explodes. Because sometimes, yes, it's really tricky to go into these things. So sometimes are reasonable sort of things. Sometimes are so reasonable that it's very hard to even remotely translate it to a plausible framework. But that's exactly what I try to do, right? So I try to give them, so either it's a very specific question, and that I can have a straightforward objective answer to, or if it's not, then it's more one of those scenarios like, we want this to happen with a plot, we want this to happen to a character. How do we make it happen that is scientifically viable, right? So one thing that I try to do is usually, either follow the line that they want, or if that line is totally out of hand, I try to come up with alternatives and saying, okay, if I understand the way that you want to go with a plot, how about you try this other event, this other type of technology, or this other type of neuroscientific happening instead of the ones that you're doing. But in the same way, always being aware that it needs to fit a general drama, it needs to fit the character, it needs to fit the plot line. Otherwise, it's not going to work. And of course, depending which kind of show, for Fringe, for instance, they really want very vivid visual correlates of things because it's a big part of the show. Other shows, for instance, there's another show that I've been working with, a fantastic writer, Alison Feltis, that was a very different process. I got involved from the first treatment, from the first script, and it's an issue of building everything. So in this case, one of the characters is actually a neuroscientist. So it's kind of along the lines that we talked about. She wants to know everything, like, what would be this person's daily life, which kind of anything, like how would you react to this, which kind of language would he use or she would use, how would you react on this situation, also in conjunction with how the science would be. And in this case, it's not a sci-fi issue, it's more of a drama kind of plot. So you don't have that much need. We also want the visual correlates, but it don't need to be as gory or crazy. So we can put more real, plausible science on it, but still opening, all these doors with it. And one thing that for me I think is actually really funny in a lot of times is that they come up with whatever idea based, on just their pure imagination, and sometimes that works, sometimes that doesn't work at all. But a lot of the times that we actually give them other options who are scientifically viable, they're actually even cooler or more interesting than the original ideas, and they love it. And they think that's fantastic. We never even knew that we could stretch it this far. And what in their head as writers is stretching it too far is actually for us sometimes a reality as scientists. So the interaction for me at least is very interesting and it's very rewarding. But again, it's going to be extremely modulated by the type of production, by the style of each writer, by what they want to get out of you. So it can go, this will be like the two extremes of the spectrum, right? So in some cases it'll be just like very direct questions and you don't really have much influence on a general script. In another case, we actually want you to the whole creative process. So to build up the characters, to build up the script, to help them figuring out where the storyline should go. That for me, it's the most interesting one, is where I get much more involved in the most interesting one. But of course, I'm giving you the two extremes of the spectrum. In most cases, it's going to be something in between.
Well, sure. And the program's basically a year and a half old at this point, and we're kind of still selling it a little bit to Hollywood. I mean, the response has been fantastic, and like you say, I mean, once they find out that, science can do a lot more than just kind of wagging their fingers and saying, oh, you silly Hollywood people, you're wrong. Exactly. That could never happen. There's a lot of people that do that, and what we try and do is say, okay, that's actually not constructive criticism. Constructive criticism means, well, no, you can't do that, but here's a suggestion. Maybe you might try this. And what more often than not they find is that the real science gives them a whole new innovative direction that wouldn't have occurred to them and can open up better storylines for them.
Exactly. I mean, that's exactly what happened. And I think you mentioned a very important part of it. It's like, for us as scientists, right, we came from a different background and from a different kind of framework, right? We've been trained to be very objective, to be very precise, to be very careful with which word that we write in a paper or we present in a lecture, because it needs to really mean exactly that. That's where we came from. That's our framework. But we need to sort of relax a bit and realize that when we move into this media kind of world, we're trying to get as much science as we can out of it and make it interesting and hopefully get the general public interested. However, we cannot treat it in the same way that we're evaluating a scientific article and say, oh, you can't do this, you can't say that. Because they're not trying to do a documentary. They're trying to do a fictional show that we're integrating some of the ideas. So as scientists, we need to be, one, open for that, and secondly, really, as we were discussing, trying to give them alternatives and other options. And it's also very true. Like, sometimes it's funny. Like, it happened on both cases, on Fringe and on this other case with Alison Feltis and other places that I've collaborated with, which is like, you give them one or two ideas and say, oh, actually, I'm going to use that for the next episode. That's actually very cool. I never thought about that. So we'll solve this one with the first way, but then the second idea, I'm going to use it in another episode. So absolutely, I think it's when it's done right, let's put it that way, I think is a very symbiotic relationship. I think it's pretty much a give and take in both ways. And I think for me, at least as a scientist, it's really interesting. It's like this deconstruction of the ideas, looking at it in a different way that I find appealing.
Yeah, and that's also the other thing I like to emphasize about the exchange is that it's a two-way street. It's not just scientists coming in and putting Hollywood straight, but in fact, the point we try and make is that scientists can actually have a lot to learn from Hollywood in terms of how to tell a story, how to construct a narrative, how to communicate effectively.
Absolutely. And I think it is a fantastic channel of communication, right? I mean, we worked together in a panel the other day on the San Diego Science Festival, and we've been discussing this very same thing, which I think is very important, is, regardless how many documentaries you try to put out there to educate the general population about, different scientific topics, regardless how many public lectures we give or you put on the news on a newspaper whatsoever, you do understand that, and we're very clear about this, that a movie, one successful TV show reaches millions of more people on that one. So if we can use that as channels to promote science, and I think that's one thing that you've been promoting beautifully with the Science and Entertainment Exchange, just trying to start building that in there, even if sometimes, of course we don't want to support something that, it's going to, in whatever scientific topic or issue, is going to put it in a completely derailed way or in a very negative light, in negative in the sense of really not being realistic and creating of issues. We don't want that. But if to some extent we can get most of the ideas out and channeled to the public in a way that, as you said, Hollywood knows how to steer its story, knows how to captivate the public. So if we can use that to channel proper science information, I think it is a fantastic channel to use. And one of the issues is that we went from a society where the general public knew nothing about science for many, many centuries to we getting this with the media boom, we get this explosion of scientific knowledge, and also because, science is cool and it's hip these days and people are interested. So, but we have a lot of cases of really bad experiences with the whole genomics boom, with the whole genetics part, and now we're having some with neurosciences, which is that we went from no information to the public to all this explosion of information, but then it's not even done properly. It comes from newspapers, from here and from there. They don't want to be sensationalist. They want to, hit their sales, increase audience rates, whatever. So it's not even done right. So it's this combo between things. So if we can use the incredible opportunity that is throughout movies, throughout TV shows, and start, steadily educating the public on that level too, I think it can be, 10 times more productive and efficient than, the other documentaries, which obviously have their place as well, but don't even by far reach as many people. And like we were discussing earlier as well, it's like if they see something in a show or in a movie that obviously is not going to be portrayed in any great depth or extent, but if it piques their curiosity and gets them interested, then they can, and go Google the subject or look for a documentary about it, look for more scientific readings about it. And if you can attract the public that way, I think will be, our mission will be great already, I think.
Yeah, I'm one of those people that compulsively Googles things that I see on TV. And the most memorable was an episode of Law & Order Special Victims Unit. I'm trying to think of a tasteful way to talk about this, but I'm just going to go with it. Essentially, there was a woman who was harvesting sperm of getting men drunk and harvesting their sperm and then impregnating herself so that she could have some sort of really super baby. And one of her victims was a very high-end surgeon or doctor of some sort. And at one point, the character is basically saying, well, if I was unconscious, how shall I say, perform? And it turned out to be a bizarre animal husbandry technique where you essentially have an electrode of some sort and you put it in a very sensitive orifice and activate it, and apparently this will cause the males of the species to ejaculate. Right. And it was real. Yeah. And it was so cool, but it was real.
Yeah, and I know that, I know that they use that in different animal big production companies, for reproduction systems for bulls, horses, etc.
Yeah.
So there's this all technology that we hopefully don't want to be changed into another scenario.
Yeah, and also I think that sometimes maybe particularly with earlier shows or older shows, what was impossible like 50 years ago is maybe it doesn't look exactly like the science fiction from the 1950s, but we have some version of that with real technology. You know, Star Trek, we have like, you know, those little handheld communication devices, our cell phones.
Right.
And I was thinking about this, and this is actually going to tie into a paper that you emailed me, because I was a big Buffy and Angel fan, and I wrote the physics of the Buffyverse, and one of the characters, Faith, the evil slayer, had a very bad habit of, like, you know, falling into comas. And in one of them, in Angel, I mean, it became this whole adventure inside her mind. Right. And what she was actually experiencing inside her mind, even though on the outside she was in a coma, was very vivid and very dramatic. And the paper that you sent me seems to be saying that actually there's some very interesting things going on with vegetative patients. We don't really understand what's going on with patients who are in that state, but apparently there's more brain activity than we might have thought.
Right. I mean, it could be. So yeah, I sent you that paper. I thought it would be interested, and we talked about the subject before, and I think it's, again, one of those, you know, it's very preliminary, but I think it could be potentially one of the great insights recently. But so it's actually from Steve Loris and Jean Picard, and they've been working on this issue of vegetative patients, what we call minimally conscious patients, for a long time, which is sort of different from coma patients, where we really believe that, you know, the conscious activity is none or very close to that. But
yeah, and actually that's a good point to make, because I think people like me, we conflate those two.
Right, we usually lump them together. In public opinion, we usually talk, you know, indiscriminately about a coma or a vegetative state patient, which is not the same. And this is actually entangled. I've been doing more and more research on this as well for this collaboration with Alison Feltis for the drama show that we're working on. But yes, I mean, but one of the interesting things is this different levels of awareness on vegetative state, right? Even on the minimally conscious patients. So even though I think they bring a very interesting, well, I should start with the caution kind of issues before anything, which they're very careful actually of the paper in saying that, which is, I think they did about 50 to 60 patients that they used and analyzed it on, and they only find it in like 5 or 6. So it's a really, really minimum percentage of patients who actually seem to have some sort of a more conscious level, even though they're in a vegetative state. So the majority does not seem to, at least as assessed by the way that they're trying to do it, right? So that's an important issue. For instance, patients who suffer from severe anoxia, so lack of oxygen, doesn't seem, the brain damage seems to be so extent that even that doesn't seem to occur, this level of minimum consciousness. But now, without the precautions, in these few cases that they actually found to be something. So what they did is actually ties up to what we thought before about training, you know, the machines or training patterns of brain activation. Now, instead of thinking about, you know, training to a category of cars and a category of food and another one of animals, is this more broad and abstract categories of yes and no, right? So if I'm going to put you in a scanner, for instance, as a subject, and I'm going to give you a lot of information that I don't want you to respond to, but I just want you to think about if your answer will be yes or no. And I can give you a ton of facts that I know that is yes, right? Is your name Jennifer? Do we live whatever? So a lot of facts that we know is going to be yes. And at the same thing, I can train, again, the computer to do it in a lot of times that you say no. And the idea is that then you can do the same thing with these patients and do a lot of facts while they're in the scanner, so while we're imaging their brain, and do the training for yes, the training for no on things that you know are known facts to you and to them. And then you can establish the pattern of activation for yes, the pattern of activation for no, and then try to ask them questions and figure out, or infer, I should say, their yes or no answers based on that. And it does seem that in this small subset of patients, the patterns of activation do reflect a coherent yes or no responses. So it does seem like an incredible breakthrough of a way to actually communicate with these patients. This is actually a follow-up of a series of studies they have done before. One of the previous cases, what they have done is, for instance, if we put a subject into the scanner and ask him different tasks, like, for instance, imagine yourself playing tennis. Now imagine yourself, reading text A, or so forth. And they look at that brain pattern of activation in normal, conscious subjects. And then they did the same thing in one case of a vegetative state patient. And in her case, the brain pattern of activation was very similar to the one that you see in conscious people, which sort of start leading them to this idea that maybe there is really some level of consciousness that is still present in there that we could try to interact with, and we try to get at it. And I think years later, or a few years later, that's what we've done with this yes and no paradigm. But I think it's an amazing breakthrough if there is in fact a way to communicate to these cases. I mean, it will be sort of the equivalent of what we talked earlier for the locked-in syndrome patients, which are totally conscious but locked into this body. If we can sort of transfer that also for these cases of vegetative state patients with some level of minimally consciousness, that we could use the fMRI as a tool to probe and to communicate with them would be fantastic. But it's still very, very preliminary. This is very exciting. It's a really hot topic. But it's one case, one study. We will need to, and we need to understand, because again, the inferences, like we talked about it, about how we interpret the brain activation that the fMRI is doing it and classify it as a yes or no. It's a lot of inferences, a lot of indirect recognition of different things. So it's tricky and it's very preliminary, but I think it's very exciting stuff.
Right, and I guess, we're about ready to wind down in about 5 minutes here, but when we're talking about something like this, I can't help thinking of the Terry Schiavo case.
Right.
And, people who hope against hope that their person isn't, that their loved one isn't in a coma or is going to wake up one day and that sort of thing. And, could this be misconstrued for political gain or to make a political point?
Right. And I think that's one of the risks of these things, is that it could, and it's a very delicate subject. It's a very delicate issue. I mean, it happened more than once to me that when we're talking about these things, in public lectures and in conferences, and talking about this idea of vegetative state patients, that you have, family and friends of patients on this state coming to us and saying, oh, so do you think that, although we have no response and she seems to be either in a vegetative state or in a comatose state, we shouldn't, cut off life support, they're conscious in there. And the truth is, in most cases, as far as we can understand, as far as we can assess it at this point, it doesn't seem to be. This seems to be much more the exception than the rule in some of these cases. And it is, again, like you said, it's very tricky because it can be used in these different ways for a different political agenda, whatever we want to build up onto it. And it is very tricky because we're walking on very thin ice, let's put it this way. I think it's great insight, and I think it's really helping us understand, or trying to understand, what could be happening in some cases with these patients. But we need to be extremely careful in how we analyze it and how we expand this type of studies. I think it shows an avenue to go as researchers that we should really explore and we should really expand to try to hopefully at some point get to those kind of answers where we actually know what's going on at different stages, at different levels of awareness. But I don't think we're there yet or close to it.
Right. And I think that, it's also important to point out that, you could tell which of the patients had that rare response, correct? I mean, it wasn't like you were misreading. I mean, there were only a very handful that had any response whatsoever.
Right. It was really a minority. I mean, one thing that is interesting is that, yes, in most cases, at least my understanding of the paper, of the article, is that there were no clear behavioral responses that could have triggered this, that would lead you to think that in that case there would be. Which sort of leads to this idea that maybe, using this as, an additional assessment, on patients to try to figure it out, it could be used potentially in the future as an additional tool of diagnosis to try to assess the state of these patients, being aware that in most cases nothing is going to come out of it. However, then it's complicated because it's an expensive technique. It's, logistically, the fMRI and get the patients there and everything is not something, that we can easily transport, like the case of the EEG that we talked about. So it's much more portable technology. So it's not an easy thing. It's not an easy diagnosis to be made. But I do think that in some cases, one of the avenues that I see this going is we could potentially start in the future using that as one amongst other ways to try to assess the conscious level of a patient. And I think from that standpoint, we should have everything that we can take to increase our knowledge about what's going on so we can make an informed medical decision, or in most cases an informed personal decision that the family would have to make in each case.
Yeah, because that's tough. I mean, you don't want to give up and you don't want to pull the plug on someone if there's any chance that they might wake up, but you have to be realistic at some point.
Exactly. And you have to. And then, of course, it's so hard and so tricky to generalize any of these things, right? Because each case is one case. It's going to be very dependent also in the rest of the physical condition of the patient. If he's in any kind of pain, if he or she is any kind of discomfort. conscious or unconsciously, all these things need to be taken into consideration. So I think it's the kind of things that each case needs to be, assessed on its own, and it's very hard to standardize. And in most cases, it really, as far as we know right now, it really does seem that in most cases, with deep vegetative state or comatose patients, it doesn't seem to be much of a possibility today of recovery. It might be with new technologies, with new therapies, with new methods. I mean, that's one of the things that we keep working every day to try, if we can actually understand the brain as a normal functioning, system, then we can, start really expanding and building all sort of psychiatric, neurological tools to deal with all these different disorders that flagellate our society. But we need to get a firm understanding of the brain as a system to explore it in that way.
Right. And I think that kind of takes us to our hour, but
I think so.
21st century era of the brain, would you agree?
I'm sorry?
21st century era of the brain.
Absolutely. I really think so. I think it's definitely, neurosciences are definitely on a top-notch position these days, and the brain, I think, is the great rock star of today's scientific field. I'm obviously biased, I'll admit it, but I do think so.
But anyway, I appreciate your doing this.
Thank you very much.
I'm sure that I'll be seeing you at some point. I
Absolutely.
seem to be going down to San Diego a lot these days.
Yes, I'm sure. Between me going to LA and you coming to San Diego, and I'm sure that we'll be working on more projects. So one of them is the stuff that we talked about figuring out the best way to integrate scientists and writers and everything. Are we going to juggle this idea of the science integration into the media world? So I'm sure I'll see you soon.
Yeah, absolutely. And I will talk to you later. Have a good night.
Thank you. Good night, Jennifer.
Yeah, bye.
Bye.